welcome again for our council seminar series. This is number 4, and our presenter today is Dr. Naufal Al-Akhsasi. So his presentation is about TILIN by target exome capture sequencing in soybeans as new and effective platform for gene functional analysis. So I hope that the presentation is about 40 to 45 minutes, and we will keep 15 to 20 minutes for questions. Naufal, thank you. Go ahead.
Hi everyone. Thank you, Abdulmajid, for your invitation. So I'm Naufal Al-Akhsasi. I'm associate scientist. I'm working at SIU with Dr. Khalid Meksem. Today I'm going to present the TILIN by target exome capture sequencing, or what we call TILIN by sequencing plus in soybeans as new and effective platform for gene functional analysis. So why concretely we work with soybeans? It's the most important oil seed crop worldwide. It's the largest source of animal protein feed, and also the second largest source of vegetable oil in the world. It's a growing market with $38 billion in revenue. This is the last data we have from 2014. Of course, right now it's much higher than that. This is like much higher than the gross domestic product of 100 countries over the world. Just to give you an idea, like 100 countries, I was looking at the statistic last time, their GDP is lower than $40 billion. So this is really a huge market in the US. The first producers are US, Brazil, and Argentina. In 2016, over 83 million acres of soybeans were planted in the US. So always farmers, they have the choice, since both of them, corn and soybeans, they grow in the same climate. So they have the choice to choose between soybeans or corn. Most of the time they do rotations just because that nitrogen in the field. So soybeans are composed by 36% of proteins, 19% of oil, 19% fibers, carbohydrates, 30% moisture, and 4% minerals. So as you can see, it's very rich. It's a complete diet between brackets. Of course, the soybean community and the farmers, they are trying to increase what we call the added value soybeans. For example, for carbohydrates, we are trying to increase the good sugars, especially the monosaccharides like sucrose versus like non-desirable polysaccharides like stachyls and raffinose. Proteins, we are trying to increase that percentage to more than 45%. And today I'm going to talk about the oil and fatty acid portion. To increase healthy fatty acids, especially I'm going to focus on the oleic acid and stearic acid. We have used 2 different approaches, reverse and forward genetic approach. I'm going to talk about the old-fashioned way, the conventional TILIN, what we call gel-based TILIN. And then I'm gonna talk about the new developed technology, which is the TILIN by sequencing plus, or TILIN by target capture sequencing. So the gel-based TILIN approach is good for, it's a high throughput screening of mutagenized populations. It's a reverse genetic approach to find novel variants in non-sequenced genomes, in polypid genomes, and also like promoter regions, exons, introns. It also reduces the number of samples for phenotyping, increased mutation rate, and this is very important, no introduction of foreign DNA, especially if you want to commercialize your crop in Europe, where they have like restrictions for importation, like they don't like GMOs. For detection of SNPs in complex traits, and also for increased number of target loci. So this is a slide summarizing the gel-based TLN. We use a chemical in our case, which is the EMS, ethyl methasulfonate. We metagenize the M1 seeds. We soak the seeds overnight on the EMS, and then we plant them next day. We to have the M1 plants. Then we harvest the M2 seeds. We plant them to get the M2 plants, and then we extract the DNA on the M2 generation. So then we go to the next step where we make 8 pools of DNA per well on a 96 well plates. We use our primers to target our gene of interest, of course, in a simple PCR. Of course, then we have what we call a mixture between homoduplexes and heteroduplexes. So the hetero, like, are like you have the wild type allele and you have the mutated allele. So that SNP there form the heteroduplex is going to be recognized by endoenzymes, especially endonucleases, and then they're going to digest that band. You're going to have, in this case, 2 double band, and then we can visualize this on a denatured electrophoresis. Of course, when we find our positive true mutants, remember that those are pools of 8 DNA, so we go back to the pool, and then we sequence the 8 individuals by target sequencing to find the real mutation, okay? So this is a summary, like quick summary of the gel-based TLN. We use this technology to clone the first gene, which is the serine hydroxymethyltransferase at RG4, which is the gene for resistance to soybean cyst nematode, which caused over $2 billion loss in the production of soybeans in the US alone. We also try to use the gel-based TLN to pool mutants on key enzymes on the fatty acid biosynthesis pathway. However, I'm going to explain why we failed to do so. I'm going to explain it later. And then we had to use other alternatives, either the forward genetic screening or the TLN by sequencing plus technology. So this is the fatty acid biosynthesis pathway. We have the sucrose. Once it reaches the plastid, it's going to be converted under the action of the ketoacyl synthase 1 to palmitic, which is the 16 saturated fatty acid. Then we have the Cas2, going to make the elongation. We're going to have the 18 saturated fatty acid, which is the stearic acid. The stereoacyl desaturase, which is the first desaturase, going to act on the stearic acid, going to add a double bond to produce the oleic acid. Then we have the Fat2, going to add another double bond to produce the linoleic acid, and Fat3 going to produce the linolenic acid with 3 double bonds. So those are the unsaturated fatty acid, those are the saturated fatty acid in the soybean genome. So first I'm going to talk about the Fat2, how many, we have 2 subfamilies. We have the Fat2-1 with 2 members, A and B. We have the Fat2-2, A, B, C, D, and E. And the SACPD, we have 4 members, which are A, B, C, D, and E. Because of the 2 duplication events that happens in the soybean genome, like 13 and 59 million years ago, so the many genes have been duplicated in different chromosomes. Not like the case in Arabidopsis, in the case you find just one gene. He do like most of the job. So in soybean genome, it's a polyoid genome due to those duplication events. So we have 2 subfamilies of the fatty acid desaturase 2. We have the Fat2-1, Fat2-2. Fat2-1, we have A and B. Those are not isoforms, those are genes. So this is in chromosome 10 and this is in chromosome 20. The same thing for the Fat2-2. We have 5 members, A, B, C, D, and E. So because of the presence of high similarity between those gene members and also different like copies and splicing variants, each time we do a gel-based TLN, and this is we did it over screen 2,000 mutants families, which is like a considerable amount of mutants. Each time we see like a band, you know, it's turned out to be a false mutant. So for the simple reason, the high copy numbers, especially during the PCRs, because primers are not specific, so probably you have a mix between different you amplify different genes, so you end up having false mutants each time you are doing that. So then we decided to go and do forward genetic screening. We started with 1,000 to 2,000 EMS mutants. We did forward genetic screening, which is a simple phenotyping. You take your mutants, you phenotype them in this case for the oleic acid content. The oleic acid content in soybeans is about 18%. And we were lucky to see that many mutants, they had like increase of oleic acid content up to 40, 50%. We sequenced, then we pulled those mutants, we did target sequencing, and we find that those mutants, they carry mutations, missense or silent mutations on the FAT21A. We find also another high oleic mutant that carry mutations on the other member, which is the FAT2-1B. So for the SAC-PD, we did the same thing. We could not do the gel-based TLN for the same reason. So we were trying to pull the 4 members, which are the A, B, C, and D. This is the Sentinel analysis for the SAC-PD members. So we have a duplication between the SAC-PD-A on chromosome 7 and SAC-PD-B on chromosome 2. This is the duplication that happened between the SAC-PD-C on chromosome 14 and SAC-PD-D on chromosome 13. Our aim was to pull mutants in each one of the 4 SAC-PDs in the soybean genome. So as you can see, high similarity. They also conserve what we call the iron-ion binding sites. They conserve the binding site, the catalysis site. So they are similar to each other. Using forward genetic screen approach, we phenotype the mutants. Stearic acid content in the first wild type is about 2 to 3%. We were able to see an increase between 6% to 20% stearic acid, which is like 7 times increase in our mutants. We obtained at beginning 5 mutants, 1 nonsense mutation and 4 missense mutations. The mutations were located in both exons of the SAC-PD-C. We did also genotype those 5 mutants on the SAC-PD-A, B, and D, but we did not find mutations on the other members. So basically, those 5 high stearic acid, those mutants with high stearic acid content, they carry mutations just on the SAC-PD-C member. So we were not able to find the mutants on the other 3 genes. The SAC-PD-C is highly expressed. It's not a seed-specific gene, since it's also highly expressed in nodules. And this is not good for the industry, because when you have a stearic acid content more than 20% due to we have what we call non-healthy nodules with the presence of necrotic cavity. So we have less infected cells with rhizobia when compared to uninfected cells that hold rhizobia. So you have less nitrogen fixation, by the way, and then this impacts the agronomical performance of the high stearic acid mutants. And that's why the high stearic lines, the commercialization of these lines has been like delayed because of that poor agronomical performance. However, when we have like like 15, 12% stearic acid, it's still low, but you still can keep that healthy nodules. And that's why we divide them into groups. So group 1 with deleterious mutation on the SAC-PD-C, and group 2 with soft mutation, what we call soft mutation on the SAC-PD-C. So then, okay, gel-based TLN, we cannot use it for genetics. We were just able to pull the SAC-PD-C. Now we had to think another alternative. How can we pull the other members, SAC-PD-A, B, and D? So then we started this project about trying to develop a new technology, which we call TILIN by sequencing plus, or TILIN by target capture sequencing. So same way, first we identify our LD50, which allow 50% germination of soybeans. The other 50%, they have like little impact, so they don't germinate at all. So we started with 10,000 seeds. We mutagenized them overnight. We end up, of course, with 5,000. Remember, this is an LD50. Then we end up with 4,000 M2 seeds. We seed increase them to the M3 generation. We collected the M2 leaves. We end up with 4,032 EMS mutant and DNAs over 4296 plates. So the idea or the objective was to isolate SACPD A, B, C, and D mutants from the 4,000 EMS mutant library. Of course, here I'm sharing some pictures to show here at SIU how we produce our mutants. This is in the greenhouse installations in the Horticultural Research Center during the spring, especially in spring. We use also in summer, of course. And this is the field that we use here at SIU to seed increase our soybeans. This is a picture from the summer, August. And this is how we harvest them in individual bags. So we don't use any combine machine, the whole work, just hand work. We have student workers that help us with this work, master students and also PhD students. So once we have our 4,030 MS mutants, so what we did, we make a pool. Of course, sequencing 42 plates is very expensive. So we had to go with a strategy in which we can decrease the number of wells and pools, you know. So we end up pulling 24 DNAs in the 2 horizontal plates and the 48 DNAs in 1 vertical plate. We design our probes that are specific to sequence, to pull mutations on the SACPD A, B, and D. And then we next proceed with the next generation sequencing. Before that, there is a step, I'm going to talk about it on the next slide. Then filtering the data, entering the data, then SNP identification, selection of the mutations, and then, of course, when you end up with 20 mutants, phenotyping then is easy, less cost, and less time. So as I mentioned before, once we extracted our DNA, we do DNA library preparation. We proceed what we call with capture-seq enrichment. So this is an easy process in which just we, using magnetic beads, we just pull. So those are our probes that recognize our target genes. So the magnetic bead just pull and enrich our sample with the just your targeted genes. And this is very, very important to increase the efficiency of sequencing. So by using the TILIN by sequencing plus, we were able to pull dozens of mutants on the SACPDA this time. Most of the mutations, so we had nonsense mutations, we have missense mutations, which are in red. So all red are the new mutants. In green, because this is a dimer, the SACPD work as a dimer. So this is just a homology modeling of one homomer. So this is the dimerization site in green. We have also in blue the catalysis site and pocket binding substrate. So most of the mutation, they were either close or at these important sites of the proteins. That's why, of course, we get deleterious mutations. Now, we have the mutants, we need to phenotype them. So again, we went from 2% stearic acid to up to 6% in average, but some individuals, they had 7 to 8%. So this is M2. We have segregation, so average is lower, but the homozygous mutations show like stearic acid up to 8%. Same thing with the SACPDB. We went from 2% to 6% in average. I'm always talking average here at M2. Same thing with the SACPDD, which is the other member. We were able to pull dozens of mutants also. And we went from 2% to also 6% on average of stearic acid content. So, okay, so now we have the mutants. If you remember, the SACPD, so SACPDC had that deleterious effect and non-healthy nodules because of its high expression in nodule. However, those members, they have high seed expression, which is good, but also they have low expression in nodules. So the question is, how this is gonna impact the nodule structure, okay? So we did the sections, microscopy sections, and as we expected, all the mutants, unlike the SACPDC, so A, B, and D, they don't impact the nodule structure and morphology. So we have high stearic acid content, and we keep that healthy nodules, which is very good for commercialization and for the soybean industry. We did the localization of the SACPDs. We find that the SACPDs, they are located on the chloroplasts. And this is a cartoon deciphering the soybean seed fatty acid biosynthesis pathway. Now we know that we have 4 members, A, B, C, and D, that convert the stearic acid to the oleic acid. Of course, the new identified members, A, B, and D, they present advantage to have like healthy nodules, so good agronomic performance when compared to deleterious mutation at the SACPDC. So currently, we are using the Telomere sequencing plus. We are pulling mutant now on the Cas2 for characterization functional genomics and characterization of this gene family, which is A and B. We also have good data about the FET2-2 and the involvement in the oleic acid phenotype. And also we have mutants on the FET3 gene family. So basically we have a mutation saturation of the soybean oil pathway. We're able to pull dozens, we have sometimes 40, 50 mutants per gene for further characterization. So I would like to thank the principal investigator, Dr. Khalid Meksem, also our PhD students, master and student workers. Joe, right now, he left, he is in Canada for a PhD. Usama is finishing his PhD. Dunya, she started her PhD, now she's in her second year. No, she's finishing her first year now. And Abdel, he's a master's student. Yeah, this is a picture from the FET conference with Dr. Meksem and Dr. Majid. And that's it. If you have any questions, I will be happy.
This is easy, so everybody can use it. In Morocco, they have, for example, lots of crops, a lot of plants where they need, to increase the genetic diversity, or they need to improve certain traits, so they can use it there too. So is it easy to use, or is it something that's very hard?
It's easy to use. We have used it with rice, we have used it with melon, with the tomato, with Arabidopsis, with the mustard. It's with wheat, so it's working fine.
So it's only taking the seeds and putting them together with the EMS, and next day take your seeds, wash them, and then plant them. That's it. So why they don't do it in Morocco? Especially they have lots of people in every lab. They have lots of students. They have a lot of, the problem here in the US is you don't have a lot of people that work with their hand, and you pay them a lot. So in Morocco, so why they don't do it?
If that's phenotyping, I mean, if they are interested to increase certain traits, like you mutagenize, you get your mutant, you simple phenotyping, they have HPLCs, they have GC machines. They don't need to do genotyping, because the most important is to phenotype those mutants and commercialize like high desired traits. So I don't think this is an issue when it came to phenotype. Maybe genotyping, that's another story.
So this would be a good project to collaborate with the Moroccan universities or institution. They can produce the mutants. They can do the phenotyping with GC and HPLC machines and other stuff that they can do very good. And then the genotyping, sequencing could be done outside. So why they don't do it?
We can propose that in the mailing list and send an email if there are faculty who are interested. We can send them to you. You are an expert in in dealing by sequencing.
That's the whole purpose of these seminars, is to encourage collaboration, and so why not send it to everybody as an invitation? There is a lot of people that work, for example, on legumes. And they need genetic diversity, so those stuff can be done, easily together with Moroccan colleagues.